Welcome back to Kidney Week. ASN thanks Vera Therapeutics for support of this plenary session.
In the heart of every hospital, there's a team of dedicated professionals. They are the experts in life-sustaining functions and the leaders in kidney care. The field of nephrology is evolving like never before. With groundbreaking innovations and scientific advancements, we're transforming kidney care. Imagine a world where augmented reality guides surgeons with pinpoint precision, ensuring the highest success rates in kidney transplants, where wearable technology and AI-powered apps continuously monitor kidney function, providing instant feedback and personalized care plans. Early detection and prevention are now at your fingertips. Gene therapy is rewriting the future, offering hope for genetic kidney diseases. Our researchers are unlocking the secrets of our DNA to develop treatments that could one day cure chronic conditions. Home dialysis is becoming more accessible and user-friendly, giving patients the freedom to manage their treatments in the comfort of their own homes. Our commitment to education is unwavering. Advanced training with virtual reality is equipping the next generation of nephrologists with unparalleled knowledge and skills. Because at the core of every innovation, every breakthrough, is our dedication to improving lives. We're not just treating kidneys, we're empowering people. Join us as we step into a new era of nephrology. The future is bright, and it starts now.
Please welcome ASN Past President and Award Selection Committee Chair, Dr. Michelle Josephson.
I'm pleased to present this morning's Lifetime Achievement Awards. The Barbara Murphy Award honors leaders who strengthen the foundation of nephrology while advancing the field through innovation, creativity, inspiration, and tenacity, and have the courage to forge new paths, overcome challenges, and serve as exemplars for future generations of nephrologists to admire, emulate, and amplify.
Barbara Murphy was astonishing in so many ways.
She transformed kidney care not only through her tireless advocacy, but also her innovative research.
She's transformed the field because she's inspired people. The Dr. Barbara Murphy Lifetime Achievement Award celebrates individuals who are pioneers in the field of nephrology, who have really done breakthrough things.
Some of Barbara's earliest work identified RNA signatures that can predict whether or not a transplant is going to do well or not, and whether or not physicians should intervene early.
Barbara is a true trailblazer in every aspect of the word. I think one amazing example is her advocacy back in the mid-90s to make sure that HIV patients had equal access to getting a kidney transplant.
Her public policy work in getting the immunosuppression bill passed will affect countless individuals for years to come.
Barbara was one of the incredible bright lights of women in leadership positions as chair of the Department of Medicine at Mount Sinai, and then a counselor of the American Society of Nephrology, and would have been its president.
She was also a great administrator. She knew how to work with people and how to move a program forward.
The first thing that you really notice about her is her smile and her warmth. She breathed life into a room.
She was, above all, a wonderful human being, a friend, a mother, a colleague, a mentor, a role model, somebody who inspires each of us every single day.
The American Society of Nephrology presents the 2024 Barbara T. Murphy Award to Dr. Vesna Garovic for her visionary leadership, research, and commitment to education in pregnancy-related kidney disorders and hypertension. Dr. Garovic is a professor of medicine at Mayo Clinic College of Medicine and Science in Rochester, Minnesota, and has a joint appointment in the Department of Obstetrics and Gynecology. She holds the Penske Foundation Endowed Professorship in Clinical Medicine and is chair of the Division of Nephrology and Hypertension. She also serves as Associate Vice Chair of Research in the Department of Medicine, Director of the Center for Clinical and Translational Science, and Dean for Clinical and Translational Science at Mayo Clinic. A native of Serbia, Dr. Garovic obtained her medical and PhD degrees at the University of Belgrade. She earned a Master of Science in Medical Genetics from McGill University in Montreal and completed her internal medicine residency and nephrology fellowship at Albert Einstein College of Medicine in New York. She has been a faculty member at Mayo Clinic since 1999. Dr. Garovic is recognized around the world for her expertise in women's health, particularly her study of cardiovascular and kidney disease in pregnant women. She and her colleagues are specifically interested in the pathophysiology of preeclampsia and the role the dysregulation of podocytes may play in the renal injury and proteinuria in preeclampsia. Her research also helps to identify epigenetic mechanisms that may play a crucial role in normal and preeclamptic pregnancies. The Garovic lab is developing tests that can help predict preeclampsia long before symptoms appear. Her team is also examining potential associations between hypertensive pregnancy disorders and future renal disease, cardiovascular conditions, and cognitive impairment. In addition to her innovative research and leadership positions at Mayo Clinic, Dr. Garovic has demonstrated her dedication to kidney disease and women's health with numerous publications, presentations, community initiatives, and tireless work for professional societies. She has served in numerous capacities for the American Heart Association and was recognized for her efforts with the prestigious Marvin Moser Award. She has also been honored for her excellence in medical practice, education, and mentorship with several awards from the Mayo Clinic. Her service to ASN has included presenting her work at Kidney Week, reviewing abstracts, and moderating sessions at meetings. Dr. Garovic and her husband, sweethearts since high school, enjoy traveling, skiing, and spending time with their family and four-legged babies. With the support and admiration of her family, friends, and colleagues, Dr. Vesna Garovic continues to advance nephrology and women's health and serves as an exemplar for future generations.
Please join me in welcoming this year's recipient of the Barbara Murphy Award, Dr. Vesna Garovic.
The Dr. Josephson Selection Committee, colleagues, friends from Mayo Clinic and from around the world. It is with the deepest sense of gratitude and humility that I accept this year's ASN Life Achievement Award. To have my name associated with late Dr. Barbara Murphy, a role model to many, including myself, is an exceptional honor that I will cherish for the rest of my professional life. There are certain conditions in nephrology that occur at the crossroads and intersection between nephrology and other specialties. I have committed my research and clinical career to obstetric nephrology, to research and clinical work, taking care about these patients and doing the research in hypertensive disorders and renal disease in pregnancy. My laboratory contributed to overall and overarching efforts of the ASN scientific community, focusing on studying the etiology, pathogenesis of preeclampsia and long-term consequences. Preeclampsia, indeed, is a disease that is still a leading cause of maternal and fetal morbidity and mortality globally, and so far we don't have any specific treatment. During my career, I had a really great honor to be mentored by some amazing professors and leaders back home at University of Belgrade in Serbia, at McGill University, Albert Einstein College of Medicine, and finally at Mayo Clinic in Rochester, Minnesota. In addition, I had the honor to serve as divisional chair to Division of Nephrology and Hypertension at Mayo Clinic, and to work together with some of the most talented, inspiring physicians who have absolute commitment to the excellence in patient care and in providing on a daily basis state-of-the-art care to our patients. I'm grateful to all of you and happy to see you all today sharing this award with me. I'm also grateful to my patients who have entrusted their care to me. Finally, last but not least, to my family, my late parents, and to my husband, my high school sweetheart, for their unlimited support and unconditional love. Finally, and in closing, I thank the selection committee, and I thank Drs. Carl Nath, Peter Aronson, and Phyllis August for nominating me for this award. This is really one of the highest achievements that I had so far, and it will serve as a constant reminder about the work that lies ahead that we need to complete. We need to continue to push the boundaries, to face the challenges, and to innovate within the space of women's health, and also in the spirit of the award and what Barbara Murphy left us as a legacy to support women in their careers and academic achievements. Thank you very much.
Our next award is the Belding Scribner Award. Established in 2006, this award honors individuals who have made outstanding contributions that have a direct impact on the care of patients with kidney disorders or have substantially changed the clinical practice of nephrology.
Colleagues around the world had great respect for Dr. Scribner.
Patients who all died now could live.
He made a profound difference on the lives of millions of people.
Dr. Belding Scribner embodied everything we strive for in clinical research. His accomplishment was to change the entire paradigm of end-stage kidney disease into a treatable disease.
He woke up one morning with the idea that he should develop a shunt.
We connect the high-pressure artery with a low-pressure vein so that the blood runs through very quickly.
In March of 1960, with the Scribner shunt, patients were now able to live.
My early dialysis sessions were done in the basement of Eklund Hall, and we would dialyze overnight on the Kiel dialyzer.
Dr. Scribner was instrumental going back to Washington, D.C. to eventually get funding for end-stage renal disease.
It really was a miraculous kind of thing to be able to come and tell the Congress about. We now have the ability to save lives of thousands and thousands of people for whom kidney failure was a death sentence.
Belding Scribner was the first chief of nephrology at the University of Washington, and also the founder of the Northwest Kidney Centers, a not-for-profit dialysis provider.
We now have a Kidney Research Institute, which is doing exciting work to bring advances to the field.
Dr. Scribner won the Lasker Award. He impacted medicine in such a profound way.
He was a miracle for patients. Without him, I wouldn't be here.
The American Society of Nephrology presents the 2024 Belding H. Scribner Award to Professor Claudio Ronco for his seminal contributions to the care of kidney patients and the clinical practice of nephrology. Professor Ronco is the director of the International Renal Research Institute of San Bartolo Hospital in Vicenza, Italy. Prior to his retirement in 2021, he also served as director of the Department of Nephrology, Dialysis, and Renal Transplantation at San Bartolo, and held the title of full professor at the University of Padua. An Italian native, Professor Ronco graduated with a medical degree and specialized in nephrology at the University of Padua, and later specialized in pediatric nephrology at the University of Naples. He has held positions as an assistant and associate professor at the Division of Nephrology in Vicenza. In the 1980s, he established a connection with his American colleagues as a fellow at Mount Sinai Hospital in New York. He returned to the USA in 1999 as director of the Renal Laboratory at the Renal Research Institute of New York, professor of medicine at the Albert Einstein College of Medicine, and dialysis director at Beth Israel Medical Center. Professor Ronco has developed novel technologies that have greatly impacted patient care. These include a revolutionary system for continuous renal replacement therapy for infants, known as the Carpe Diem system, which is now in use in Europe and the US. He has also been instrumental in the development of technology for hemodiafiltration, an emerging technology for a wearable artificial kidney. Widely known as a leader in acute kidney injury, Professor Ronco founded the Acute Disease Quality Initiative and the Acute Kidney Injury Network, which have resulted in important developments in the field. Since 1982, he has organized yearly scientific courses in Vicenza to discuss critical care topics and renal replacement therapies. As the founder and director of IRRIV, he has trained and taught doctors from around the world on the Vicenza team techniques. Professor Ronco has published more than 1,700 papers, 120 book chapters, and nearly 100 books on nephrology, dialysis, and intensive care. He has delivered over 1,000 lectures and presentations. A past president of the International Society for Hemodialysis and former council member of several scientific societies, he is currently editor-in-chief of Blood Purification, Contributions to Nephrology, and Cardiorenal Medicine, and editor emeritus of the International Journal of Artificial Organs. His pioneering work has been recognized with numerous awards, including the NKF International Medal of Excellence, the ISN Bywaters Award, and the ISHD Scribner Award. In addition to his innumerable scientific and educational endeavors, Professor Ronco has long enjoyed such activities as ice hockey, skiing, boating, playing in his band, and spending time with his family. Having dedicated his distinguished career to educating and training generations of nephrologists and critical care physicians and improving patient care, Professor Claudio Ronco is widely regarded as a monumental force in nephrology.
Please join me in welcoming this year's Scribner Award recipient, Dr. Claudio Ronco.
Dear friends, forgive me the emotion. In these days, I heard friends, colleagues telling me I'm a pioneer, a legend, an icon. All these terms in common have one thing: it means I'm old. But to me, this also has a special meaning because I had the chance to meet the heroes of dialysis. Pim Kolff, Jack Meyer, George Schreiner, many people. And all these people belong to the family of ASN. I still remember my first ASN in Chicago in 1982. I was a fellow at Mount Sinai Hospital under the mentorship of Juan Bosch and Marvin Levitt. We booked a room with 2 beds, but we were 5 fellows. And we had no money, so we had to sleep on shifts in the same room. But it was a fantastic experience with Eric Tepper, Alan Lauer, Susie Liu, all the other fellows. And we presented 2 posters, one on renal functional reserve. Barry Brenner himself came to that poster and signed the poster and said, Claudio, this is the beginning of understanding that when you lose one nephron, you start the slippery slope of chronic kidney disease. So please go ahead with these studies. The other was a poster on continuous arteriovenous hemofiltration made together with Juan Bosch. And Belding Scribner came to this poster. He told me, Claudio, we always wanted to do this, but we did not have the supplies, the device. I'm so glad you did it. And also, he told me, please never give up to try to improve dialysis. You have the strength to do it and teach other people to do that. And this is what I have tried to do in our research institute, having fellows all over the world that now carry over this type of research. You know, we all as nephrologists dream a world without dialysis. But we're not there yet. And believe me, while we're dreaming about a world without dialysis, I think we should concentrate in improving our dialysis techniques, our devices, because our patients are really expecting this from us. So if I can give a message to the young fellows in the room, do not take for granted that everything has already been done. Don't accept that somebody older than you tells you, oh, forget about this, this has been done. New sciences, microfluidics, new biomaterials can help you to achieve what was not achievable a few years ago. And this is the message of Belding Scribner that I adored really for all my life and I took as a model to try to change something in the life of our dialysis patient. With this, I want to thank all the friends, in special measure, people at ASN, because ASN has always been a family for me. And I have to tell you that I received this award on behalf of all the colleagues, the nurses, the technicians that have spent their life at the bedside of dialysis patients. Thank you very much.
Established in 1964, the Homer Smith Award is presented to an individual who has made outstanding contributions which fundamentally affect the science of nephrology.
Homer Smith was a visionary.
He was curious, he was innovative.
He was foundational to modern nephrology.
The Homer Smith Award of the American Society of Nephrology was the first award given, and it reflects Homer Smith's dominant role in understanding the physiology of the kidney.
Smith led the Department of Physiology for decades at NYU, and it was Smith, probably more than any other person, who defined the modern concepts of glomerular filtration, tubular excretion, and also secretion.
Homer Smith truly inspired generations of physicians, including my own.
He brought to nephrology principles upon which our field is based.
All of these basic things inform our care of patients.
He used comparative biology in order to advance our understanding of more complex systems, the human kidney.
He brought to NYU techniques and information that he had gathered by studying fish. Many of those experiments were carried out in Mount Desert Island.
The Mount Desert Island Biological Lab played an extraordinary central role. Smith realized that marine organisms gave him powerful tools to study the evolution of the kidney.
He was into philosophy, music, science in the broadest sense, religion.
To read him, it sort of opens your mind to other areas and it fires the imagination.
When he said something, it turned out to be correct because he was very precise in what he did. And that's a lesson for all of us.
He really set the stage for all future researchers and how to move the field forward.
The American Society of Nephrology presents the 2024 Homer W. Smith Award to Dr. Joseph Bonventre for his seminal research contributions and leadership in science and nephrology. He is the Samuel A. Levine Distinguished Professor at Harvard Medical School and Constantine L. Hampers Distinguished Chair of Renal Medicine, Chief of the Division of Renal Medicine, and Founding Chief of the Engineering and Medicine Division at Brigham and Women's Hospital. He was Harvard Director of the Harvard-MIT Division of Health Sciences and Technology. Born in Brooklyn, Dr. Bonventre earned a B.S. with distinction in engineering physics at Cornell University, his M.D. at Harvard and MIT, and a Ph.D. in biophysics with Claude Lechen as his mentor. He completed postdoctoral training at Harvard and Massachusetts General Hospital. Dr. Bonventre's research focuses on the study of kidney injury and repair and signaling transduction, with a special emphasis on the role of inflammation, biomarkers, and stem cells. He has authored over 600 publications and 3 books. His work has been cited more than 96,000 times. Among his early contributions were pioneering studies regarding the role of calcium in mesangial cells and acute kidney injury, AKI, as well as the cell signaling role of phospholipase A2 and early studies of gene expression with AKI. His laboratory identified the role of sex and susceptibility to AKI, the importance of inflammation in AKI, the role of senescence, and the cells involved in adaptive and maladaptive repair after kidney injury. Dr. Bonventre and his collaborators influenced the definition of acute kidney injury. He identified a key general transcriptional inhibitory domain that subsequently enabled CRISPR-I technology. He cloned and characterized kidney injury molecule 1, KIM1, as a sensitive diagnostic and prognostic biomarker of kidney injury and progression of chronic kidney disease and kidney cancer. He and his lab have extended his kidney repair and stem cell work to create human kidney tissue models of disease. His protocols to generate kidney organoids have led to a revolution in kidney research using human tissue. Dr. Bonventre's work has been recognized with numerous honors, including the International Society of Nephrology Bywaters Award, the Osler Medal of the Royal College, and 2 Merit Awards from the NIH-NIDDK. He has been elected to the American Society for Clinical Investigation, the Association of American Physicians, the American Institute for Medical and Biological Engineering, and Fellow of the American Association for the Advancement of Science. A past president of ASN, he is currently on the editorial board of JASN, CJASN, and Kidney 360, and is editor-in-chief of Seminars in Nephrology. He has played a number of leadership roles in ASN initiatives over the years. He has been awarded honorary doctorate degrees from the Norwegian University of Science and Technology and Mount St. Mary's College. Dr. Bonventre and his wife, Dr. Christina Cannon Bonventre, have 2 children, Andrew and Joanna, son-in-law Brian, and 3 grandchildren, Daniel, Alex, and Taylor. The family, including Joe's parents, Fanny and Vincent, along with his friends, mentors, trainees, and colleagues worldwide, have long supported Dr. Joseph Bonventre in his various roles as scientist, educator, clinician, and as a foremost contributor and leader in nephrology.
Please join me in welcoming the Homer Smith Award recipient for this year, Dr. Joseph Bonventre, who will present on kidney injury and repair.
Well, that's sort of amazing. I'm really humbled and greatly honored. I want to thank the ASN. I want to thank Dr. Cruz, Dr. Josephson, and all my colleagues over the years and trainees. This is an incredible award, the most prestigious I've certainly ever received, and suspect the most prestigious I ever will receive. So now I'd like to start my presentation. Let's see, okay, and start with these disclosures. And then I want to say that, clearly this award really belongs to all my former fantastic trainees and colleagues and collaborators. On the next 3 slides, I've illustrated approximately 1/2 of these trainees. I think you'll recognize many of them because many of them have gone on to great things in medicine, in nephrology, and in other fields. I also want to recognize my mentors. As you heard, I was an undergraduate at Cornell in engineering physics, and then my life was transformed by an experience in the summer program at UCLA and Caltech and the Jet Propulsion Laboratory on space biology. And I came back from that as a rising senior in college and decided that I wanted to do something related to biology and medicine. I then applied for graduate school and was accepted by Arthur Solomon at Harvard in the Harvard Biophysics program without any biology course on my application. So I came to Harvard as a graduate student initially, and then looked around and looked for like-minded people. There were at the time two biomedical engineers at Harvard College, and one of them was Tom McMahon, who then directed me toward Ryan Bucas and Cliff Barger in the physiology department at Harvard Medical School, who introduced me to the kidney. I then, as a graduate student, evolved into someone who really wanted to do medicine also. And at that point, Irving London accepted me into this new program, which is the Harvard-MIT Division of Health Sciences and Technology, which I subsequently had a leadership position in. And my PhD advisor was Claude Lechen, as you've heard. I finished my PhD on renal concentrating mechanisms and then went to the MGH for residency, and where my research and overall mentor was Alex Leaf and clinical mentor Pete Coggins. Stayed at the MGH for 25 years or so, and then Victor Zhao recruited me to the Brigham to replace this man, Barry Brenner, whose name has already come up. And Barry, of course, is someone who can't be replaced, who could never be replaced. And unfortunately, Barry had passed away this summer, but he contributed such an enormous amount and was an incredible role model for what one person can do in an entire field. I also want to thank my colleagues at the Brigham This is one picture of the large division that I've been having the privilege of working with over the years. Many of these people who are still there or who have left and gone on for leadership positions at other institutions throughout the US and the world. I also want to recognize my other division of engineering and medicine, where Victor Zhao was at least tolerant of my idea about establishing this. This is, I think, the only such division in a department of medicine in the country. And right now it has about 140 faculty, trainees, and associates. So now let me turn to the science, and there's been a lot that I've been involved with, and so I'm going to actually present a sampling, truly a sampling, years worth of work summarized in one slide in many cases. And I'm going to mention some of the fellows. I'm going to call them out, or at least by having a picture on the slide. But of course, in anything I mention, there are many, many trainees and colleagues who have been involved. So my career has basically been associated with injury and repair in kidney, and driven by these four questions. One, how do we best identify injury to the kidney? What are the primary pathophysiological contributors to the injury? How does the kidney repair itself in an adaptive way? And why does the repair process become maladaptive? Can we develop ways to restore, create human kidney tissue and better model human kidney diseases in our constant quest to find new therapeutics for our patients? So I'll start actually with a collaborative study. Again, collaborators have played an enormous role in my career. And this is with Glenn Chertow, and actually was initiated in the airport, an airport waiting room, when the two of us were coming back from a meeting. And this study essentially demonstrated the importance of small changes in serum creatinine, 0.3 milligrams per deciliter. With an odds ratio for death, just looking across the Brigham database over a year, odds ratio for death of 4.1. And this study helped to influence the change in the way we thought about acute renal failure at the time, and led to the change. You've heard about ADKI and the role of Claudia Ronco and others in ADKI. One of the groups that came together to define kidney injury, acute kidney injury, and then subsequently the KDIGO definition. Both of these definitions took into account these small changes in creatinine so that you didn't have to get failure to really have a significant injury to the kidney that was meaningful for patient survival. But of course, creatinine, we all know, is not a great marker for acute events in particular, as reflected on the right-hand side of this slide by the relationship between creatinine and glomerular filtration rate, one marker, a function of the kidney. So now I'll move into other biomarkers and other ways to look at injury, and particularly focus on kidney injury molecule 1, which Hari Ikamura in the lab, he's still in the lab with me as a faculty member, but as a fellow, he, working with Michelle Santacola and Veronique Bailey, collaborators, was doing some studies looking for genes that could be modulated in expression with ischemia. These studies, actually, I personally did these animal studies in the rat. And so we prepared messenger RNA from post-ischemic rat kidney and contralateral kidney that was normal or closer to normal. And then at the time, this was a time of when mRNA chips were popular. That was the leading technology. But we didn't jump on that bandwagon at the time. We used a completely unbiased PCR-based method. I won't go through the details, but it was an iterative method where you amplified the genes that were upregulated differentially between two populations. And with that, we discovered a molecule which we called kidney injury molecule 1. It's a single transmembrane domain molecule, heavily glycosylated and cleaved by metalloproteases, and appears in the human urine. And that was one of the driving forces for us to continue the work, because early on, we knew two things that we found were particularly interesting. First of all, it was in the proximal tubule. And second of all, it was clearly relevant for humans. It was also very interesting to us in that it was primarily a kidney-specific protein. And then Vishal Vajja, as a fellow, showed in the rat that the levels of KIM1 in response to cisplatinum, the levels increased in a quantitative way in the urine well before there was any change in blood urea nitrogen or creatinine. So there's 1 to 2 days after cisplatinum, there were marked increases. The baseline levels were extremely low. So marked increases in the levels of KIM1 when creatinine and BUN had not changed. And fast forward, we've done a number of studies in humans and tissue, but this is a more recent one with and as a collaboration with Sandro Sandagati and Jia-Young Chen at Harvard. And this demonstrates the utility of this molecule at a time, this human biopsy, at a time when the histology was relatively mild. This was a nephrotic syndrome patient who had minimal change disease. Minimal change, hence the relatively mild in terms of abnormalities, mild tubular dilatation, focal brush border loss, isolated mitotic cells, all characteristics of kidney injury, but relatively mild. And yet you look and see the very dramatic pink color of practically all of the proximal tubules. And then subsequently working with Predictive Safety Testing Consortium, which was a consortium primarily of pharmaceutical company representatives looking for new markers to help in drug development. We looked at 10 different nephrotoxicity models established by different companies and looked at a number, approximately 10, of biomarkers. And here is a summary at the top of all of the data as a function of the area beneath the curve, of the receiver-operator curve, and with varying amounts of histopathology. So 0 to 1 meaning that there's very little histopathology in the rat kidney, and yet KIM1 performed incredibly well, the ideal biomarker being one that has a or has an area underneath the curve of 1 in the way of representing this. And certainly when there was little injury to the kidney, there were BUN or N-acetylglucosaminidase, NAG, or the serum creatinine did not really perform very well. And so this was published here, and then later that year, a European consortium came to the same choice of showing that KIM1, again, completely independently, turned out to be a very good marker for injury in the rat here. So in 2008, KIM1 was qualified by the FDA and the EMA in Europe, and also the Japanese Regulatory Society, as a preclinical safety marker for assessing kidney toxicity of experimental drugs. And actually, a number of years later, the FDA published a paper looking at the regulatory applications after this qualification, and you could see that when, at the time, the molecule was qualified. Subsequently, the number of regulatory applications that reference KIM-1 as a drug development tool increased substantially. In 2018, more recently, KIM-1 was qualified by the FDA as part of a composite to use in phase 1 clinical trials to evaluate nephrotoxicity. This was based on studies of gentamicin and cisplatin in humans. Okay, so that's urine. We're talking about urine KIM-1. Venkata Sabasetti, Sabi, when he was a fellow in the lab, looked to plasma KIM-1. So this was now around 2014, and found that plasma KIM-1 increased with ischemic injury to the kidney. Even periods of 10 minutes of ischemia or 20 minutes of ischemia, which had essentially no effect on creatinine 24 hours later, produced significant increases in plasma KIM-1, in addition to what was expected already at that time, the absolute urinary KIM-1 levels or the normalized urinary KIM-1 levels, normalized to creatinine. And in collaboration with Natalia Nowak and Andrei Krylewski, we also showed in that paper that plasma KIM-1 was a good predictor of bad outcomes, essentially, in a population of patients with type 1 diabetes. So if the plasma KIM-1 was above a certain level, that patient was much more likely to develop either end-stage renal disease. And so that led to a number of other studies. This was one we were not involved with, but this was a European study with 2 diabetes cohorts where they looked at 297 blood markers and compared those markers as prognostic of the decline in renal function. And this was adjusted for many clinical covariates. And of the 297, 2 sort of stood out, one being KIM-1 and one being member of the TNF receptor family. And here's a number of papers, or not a complete list, but a number of papers that involve or show that plasma KIM-1 is a prognostic biomarker for progressive kidney injury or cardiovascular disease or mortality. The studies in yellow were studies from the CKD biomarker consortium that I was privileged to be associated with. Okay, and another aspect that's been interesting and timely right now is that we discovered many years ago, we expected this, actually, because we knew a lot about the molecule, that it's markedly upregulated in renal cell carcinoma and in lymph node metastases. And fast forward to a paper that we recently published in collaboration with Vincent Zhu, junior faculty member at the Dana-Farber rather, showing that with increasing levels of plasma KIM-1, you could predict a bad outcome in renal cell carcinoma. And this has also gotten a lot of press at the ASCO meetings. And here I've just quoted the non-invasive biomarker KIM-1 is front and center in renal cell carcinoma investigations. And Tony Kaluri, who's the leader in this field, sent out an survey of what's the most important biomarker in renal cell cancer. And it turned out of these 4 choices, within an hour, he got 200 replies on KIM-1. Okay, now let me move on from KIM-1 to primary pathophysiological contributors to injury. So we started out, as you heard, really studying intracellular calcium at a time when this was actually the first representation of using fluorescent dyes, Quin2 and Furat2, to measure intracellular calcium both in epithelial cells of the kidney and in mesangial cells. And at the time, we also were studying phospholipase A2 because we thought that that was a primary contributor to mesangial cell injury. And this was a time when, if you did a knockout, you could get published in Nature. So you see how old these data are. Okay, and then it was mentioned on in the video that we also early on looked at the role of sex and showed that males were much more susceptible than females, and that we could introduce a male phenotype to intact female mice by giving them testosterone. And then we did orchiectomy, not on this slide, in males and converted them into a female protected phenotype. And more recently, Morgan Grams and her group looked at over 1.2 million participants in meta-analysis, and also demonstrated in humans that, again, the mouse studies were predictive of the human outcome, that females are less susceptible than males to acute development of acute kidney injury. So at the same time then that all of this other work was going on, again, we were very interested in molecular characteristics. What made the post-ischemic kidney more susceptible to injury, or what are the characteristics? What genes were upregulated? And here was Ralph Wirtschgall cloned a gene that we called KID1, wasn't known at the time, so we got to name it, for kidney injury and developmentally regulated gene 1. And that gene had a number of zinc finger motifs, and we found that it was a transcriptional inhibitor. We then mapped the region of the gene. These are the representation of the protein in different domains. We mapped the region of the gene that was responsible for the transcriptional inhibition using a CAT readout here, where you attach the part of the gene protein to to GAL4, and that binds to binding sites on the reporter construct. And then we found proteins that interacted with this domain, which was responsible for the inhibition. So looking at these here, the inhibition of transcription. And it turned out, fast forward 15 years later, that others have used this CRAB domain as a basic principle for CRISPR-I technology of suppressing transcription of particular proteins in general. We spent a number of years on inflammation, and this is a reflection of all of those years, both looking at the endothelium and looking at the role in the tubule. And here, back in 2004, we concluded that ischemic acute kidney injury, or acute renal failure at the time, was an inflammatory disease. And this has been confirmed by many others, including in a paper in Nature by the KPMP consortium. Okay, how does the kidney repair itself in an adaptive way, and why does a repair become maladaptive? Early on, again, Ralph here, showing that as part of the repair, there was a proliferative response. And in addition to proliferative response as measured by multiple markers, there was also a transition. The epithelium took on characteristics of the mesenchyme, and subsequently, in this case, vimentin. And so then we also, early on, did a number of studies on preconditioning. And here, this phenomena of preconditioning was really profound, in the sense that if you made the mouse ischemic, if you made the mouse kidney ischemic, the creatinine went up, came back down. This was bilateral ischemia. If you then came along 8 days later and took the same mouse and you increased the period of ischemia, there was no change in serum creatinine. So there's clearly a lot going on, with even persistent up to 8 days, that was changing the response of the kidney. And so that was what we were looking for, with mechanisms or processes that we could then recapitulate using pharmacological tools. But the other thing that we found at the time was, here was at a time when basically people thought that after ischemia, the kidney completely repaired, but 12 weeks later, even though creatinine had been way down pretty much by 5 days after the ischemia, there was persistent activation of myofibroblasts. And so this was a very early indication that acute kidney injury could lead to more chronic changes in the kidney. And then Ben Humphreys and Jeremy Duffield in the lab, basically in collaboration with Andy McMahon, worked on this whole issue of what cells are involved in the repair process. So we know that there's a lot of proliferation, there's a dedifferentiation in the epithelium, loss of polarity. Did the cells that replenish the lost cells come from the bone marrow? Did they come from interstitial stem cells? Or did they come from stem cells in the epithelium itself? And we actually found that they didn't come from any of those sources, but they came actually from proliferation of the surviving epithelial cells left after the cell death. And then again, looking at different mechanisms of injury here, when Li Yang was in the lab with Guillaume Queneau and Seiji Akishi and Craig Brooks, developed this whole notion of senescence, that in fact, what happened with injury was that the cells went into a cell cycle arrest period, and they then produced molecules that were profibrotic. And this shows with moderate IRI, there's a return of different phases of the cell cycle that the cells respond, that are entering the cell cycle, whereas with aristolochic acid or unilateral ischemia reperfusion injury, where there was much more fibrosis, that the cells did not respond, there was not a return, there was a persistent senescence. And this has led to a number of studies by many groups looking at senescence as a major contributor to the abnormal repair that goes on after injury, and ultimately resulting in progressive fibrosis and chronic kidney disease. Okay, let me just say that KIM1 also plays a role here. KIM1 under normal circumstances is a phosphatidylserine receptor. It's an anti-inflammatory response of the proximal tubule to slow down inflammation, as reflected by this cartoon where the apoptotic bodies are taken up because they're coated with phosphatidylserine, and this ultimately results in antigen presentation and upregulation of Tregs, downregulation of inflammatory immune cells. But more prolonged levels of KIM1 expression in studies honchoed by Yutaro Mori and Hari, this becomes maladaptive by adding to lipotoxicity, mitochondrial stress, DNA damage, cell cycle arrest, and profibrotic mediators. Okay, and then the last, I'm a little over, so there's a few more slides, please. But I want to say that essentially, how do we create, restore kidney tissue? You're going to hear a lot of this in Melissa Little's talk that's coming up. But I want to just say that in studies initiated by Albert Lamb and then greatly expanded and brought to conclusion by Benjamin Friedman and Ryuji Morizani, this is an example of the generation of kidney tissue with markers of the glomerulus, the proximal tubule, and the distal nephron in studies. Ryuji was the primary author here, first author. And at close-up, you can see the generation of what looks like Bowman's capsule around a podocalyxin positive tuft leading into a proximal tubule. And here, a proximal part with a distal part, the V-cadherin, with TAM-horsfol protein in the middle. And EM, there's very interesting structures that are formed, looking like podocyte bodies, as well as basically villi and intercellular junctions. Bino Friedman developed at the simultaneously published the same year, another protocol And he basically then has developed this, then used CRISPR technology to knock down PKD1 or PKD2 and establish this as a model for cyst formation. And he's gone on to really develop this in an incredible way, looking for new therapeutics. And then finally, in terms of from a science point of view, this was Kyle McCracken and Min Shi in the lab, who developed a technology to develop collecting duct organoids and ureteric bud organoids. And you're going to hear, I think, maybe a little bit of this from Melissa also. But this technology results in, when examined by single cell analysis in a population of cells that are primarily cells of the collecting duct, principal cells. Okay, I want to thank, again, the mentors, my trainees, my grant reviewers, who have been, when they were kind, they were, but when they weren't so kind, they also motivated me to prove them wrong. And so I thank them. And the funding agencies, in particular, the NIDDK, which has supported me over the years, and actually a lot of what I presented was based on 1R01, which is in its 39th year. And these are some of the collaborators, only ones from the 3 consortia. Again, there are many, many others. I want to thank again ASN. This is a year from my council group when I was president. I want to thank Todd. And when I was president, that was the year that we modified this this celebration from Renal Week to calling it Kidney Week, which I'm also proud of. My family, you've already seen pictures of my daughter Joanna, my son Daniel, yeah, Andrew. You get that confused a lot, and my 3 great grandsons, Daniel, Alex, and Taylor. Alex and Taylor are here. Joanna's wife, Brian, my mom, who's celebrating her 99th and a half birthday, and my dad, who's unfortunately not with us now. And of course, my wife, who's been with me the whole time, and thank you. We've just celebrated 2 weeks ago our 50th wedding anniversary. So thank you all. Sorry.
All right, to continue the proceeding. My name is Rashid Badigeshin from Duke University. In my role as the Kidney Week Committee co-chair alongside Dr. Tamara Isakova, it's been exciting to plan a meeting of this size and scope, including the plenary speakers. Today's state-of-the-art lecture will be delivered by Melissa Little on understanding, improving, and applying human pluripotent stem cell-derived kidney tissues. You've got some little things about this from Dr. Bonventre. Professor Little is the Chief Executive Officer of the Novo Nordisk Foundation Center for Stem Cell Medicine, Chief Scientist at the Murdoch Children's Research Institute, and leader of the Kidney Regeneration Laboratory in Melbourne, Australia. She's the immediate past president of the International Society for Stem Cell Research and holds an honorary position as professor in the Department of Pediatrics, University of Melbourne. She's internationally recognized for her work on kidney development and her pioneering studies into potential regenerative therapies in the kidney. Professor Little's approach to generating kidney organoids from human pluripotent stem cells has been adopted across the globe, where it is being applied to disease modeling, drug screening, and kidney replacement therapies. So if you look through the posters in the last 2-3 days, every other poster has something to do with iPSC and organoids. That's because of the pioneering work of Professor Little. Professor Little's work has been recognized by many awards, including the ASN Omar W. Smith Award in 2021 for outstanding contributions that fundamentally affect the science of nephrology, and the NHMRC Marshall and Warring Ideas Grant Award for being the top-ranked recipient in the Ideas Grant Scheme for 2021. Professor Little is a companion of the Order of Australia, Fellow of the Australian Academy of Science and the Australian Academy of Health and Medical Sciences, a Fellow of the Danish Royal Academy of Science and Letters. She's on the editorial board of many journals: Cell Stem Cell, Nature Reviews and Nephrology, Development, and Kidney International, among others. She has previously held the role of President of the Australasian Society for Stem Cell Research, Program Leader of Stem Cells Australia, and the Chief Scientific Officer of the Australian Stem Cell Centre. I am deeply honored to introduce our distinguished plenary session. Professor Little, welcome.
Thank you very much for the kind introduction. I'd really like to thank Dr. Cruz as the president of the ASN and also the Kidney Week organisers for this opportunity to be able to speak to you today. And I'm going to talk to you about pluripotent stem cells and what we can or can't do in terms of using these to actually model human tissue. So these are my disclosure statements. And I want to tell you this story in 3 parts. I want to start by telling you about kidney development, which is really my area of scientific research, and how that understanding of kidney development has helped us to build models of the human kidney. I then want to talk to you about applications and share some of the published but also unpublished work that we are doing in this area. And then finally address the question of whether you can genuinely engineer a tissue that could be used therapeutically one day. I don't really need to introduce this audience to the kidney and this beautiful pair of organs that keeps us alive, balances our fluid, and is essential for our lives. But I will just refer to the functional unit of the kidney, this incredible patterned and segmented epithelial tube, the nephron, of which on average a human kidney contains approximately 1 million nephrons. Although we've learnt from the late Barry Brenner that the number of nephrons you carry is impacted by what happens to you during development. And that's because all of your nephrons are forming as you are developing. You don't have any capacity to make new nephrons after birth. So you can injure a kidney, and Joe has told us very elegantly about what happens in response to that injury, but you cannot make new nephrons. So how then do you form a kidney in the first place? The kidney is, from an embryological point of view, a mesodermal organ, and the nephrons themselves arise from this population shown here, a 6-2 expressing stem cell population that's mesenchymal in nature and is receiving signals from the adjacent branching ureteric tip that's going to form the collecting duct to turn over, but is also able to receive signals to get a small number of cells to aggregate and then go through what we call a mesenchyme to epithelial transition to form this very early renal vesicle. This is the birth of one nephron. Almost as soon as this structure forms, there is a polarization, so that one end will become the distal end of the nephron and the other end will become the glomerulus. And so you subsequently get a patterning and segmentation such that the distal end connects to the collecting duct, the proximal end pulls in the vasculature to form the glomerular capillaries, and you get a differentiation of the many different cell types along the proximal and distal segment and the loop of Henle that give you unique functions of the nephron. But as I said, this is an event that happens during development because this progenitor population is lost in humans in trimester 3 of development. So we really want to ask the question, how can you recreate a nephron? And Joe has touched on this briefly, but our ability to do this comes from two seminal discoveries. The first of these was James Thomson in 1998, who isolated an inner cell mass from a human blastocyst and showed that he could culture these human embryonic stem cells in a dish and keep them pluripotent, that is, able to be differentiated further into individual human cell types. The second discovery was from Shinya Yamanaka, and this won the Nobel Prize. In 2006 in mouse and then 2007 in human, he identified 4 key transcription factors which you could introduce back into any adult cell, any somatic cell. It can be a fibroblast of the skin, it can be a white blood cell in a small blood draw, and take those cells right back to a starting point, a pluripotent state, essentially equivalent to that of the inner cell mass without destroying any human embryo. This again gave us a lot of hope and the potential to actually take those cells and differentiate them further. But can you make a kidney? And this is where our developmental biology is very important, because in fact the kidney, as with every other tissue, comes from the early fertilized egg. And during early development, you have to go through a process of gastrulation to form a primitive streak. The posterior primitive streak becomes the mesoderm. Along the trunk, there is paraxial mesoderm that's making somites, lateral plate mesoderm, and then intermediate mesoderm from which the kidney arises. And both the collecting ducts, which are a side branch of the anterior intermediate mesoderm structure, the nephric duct, and also the progenitors that form the nephrons are also intermediate mesoderm. For this reason, you can actually build a roadmap that says, if I'm starting with pluripotency, I need to step through each of these steps in sequence to get to the point of building a kidney. And we knew markers of each of these states. We knew a lot about the signaling factors that were required for gastrulation, for commitment to mesoderm, and essentially this was our blueprint to follow. And I'm going to cut to the chase. This was a very brave postdoc in my group, Minoru Takasato, who tried many different approaches to reach this. And then in 2014 and again in 2015 with a revised protocol, he actually showed we can take pluripotent stem cells, either ES cells or iPS cells, walk them through each of these stages, make a micromass of these cells, trigger them to go through that mesenchyme to epithelial transition, and across the next 3 weeks build a microorgan, which we call a kidney organoid. So this tissue is now a couple of millimeters in diameter. It's clearly not made up of pluripotent stem cells anymore. What you can see stained here is the glomeruli in white for nephron antibody, proximal tubule in blue, distal tubule in green, and this late distal, or what we initially thought was collecting duct, that all of the nephrons are plumbing into. So this is really quite a remarkable tissue. You can see as the antibody staining adds on that these are interconnected, they're patterning and segmenting. Here's the proximal tubule segments coming up, and you can see that they're forming glomeruli as they would during development. Around those nephrons is a stroma, a renal stroma, and also very early vascular progenitors. You can see in red this plexus that's forming. The glomeruli are forming interdigitating foot processes for their podocytes, and in fact you can sieve the entire podocyte bundle out. It's starting to make a polarized basement membrane, and Rachel Lennon has worked with us to show that these go through collagen switching to form appropriate GBM markers. These are the glomeruli, and these are perhaps the most beautiful part of this tissue. You have a claudin-positive parietal epithelial layer, the podocytes, an early GBM forming, and you can see this vasculature around it. So now we have a three-dimensional model of a human glomerulus. When we first did this work, there was no single-cell sequencing. We did bulk RNA sequencing and compared it to a panel of tissues from human developing organs and showed that these mature organoids matched kidney most closely. Subsequently, we and many others have done single-cell analysis across these protocols and have shown the appropriate kidney cell clusters are present within our organoids. So I'm going to conclude at this point that you can derive from human pluripotent stem cells a model of the human kidney that has numerous distinct cell types and evidence of nephron formation, patterning, segmentation, renal stroma, and early vasculature that transcriptionally matches a trimester 1 human kidney. So 10 years now have passed, and we've spent a lot of time really interrogating what these structures really have in them, having a look at how they can or can't model disease, having a look at what happens when you transplant them into tissue, and starting to really look at the engineering of this. And I want to walk you through these applications in the next part of my talk. But I would give a shout out to the fact that we were not alone. That, as Joe pointed out to you, there are a number of papers that came out very close together around that time with slightly different protocols, all showing some degree of nephron formation and all walking through these developmental steps: primitive streak, intermediate mesoderm to nephron formation, which really highlights the importance of understanding development. So how have we applied these in disease modelling? What can we really model with a tissue like this? I think it's important to step back again and talk about the challenge in inherited kidney disease. Many pediatric nephrologists and geneticists in the audience will recognise the fact that collectively this is a considerable problem and that there are no novel treatments for these patients, even once we get a diagnosis, which is increasing. And I think this is in part because animal models are not always good models of human, and also that immortalized and primary kidney cell lines have not necessarily helped us to find mechanism or new therapy. So what about an organoid? I point out here that the many, many genes that are mutated to give you different forms of inherited kidney disease give you different outcomes because they're frequently expressed in a very specific cell type along this nephron. And so you need to have that cell type and that gene expressed if you want an organoid to model your disease. So I'm going to start with the glomerulus because I think this is a bigger success. As I've shown you before, the glomeruli in these organoids are really beautifully patterned three-dimensional structures. Along here you can see the co-staining of slit diaphragm proteins. And if you look at the gene expression of these critical podocyte markers in an organoid glomerulus compared to the immortalized line that is frequently been used in studies of the podocyte, you can see this really vastly improved. And that three-dimensional structure is incredibly valuable to the podocytes and their functional identity. So we've actually made pluripotent stem cells from patients with mutations. This was our first one in a patient that presented at the Royal Children's Hospital with 2 distinct mutations in MPHS1. And we showed we could actually pull out the glomeruli from these and show that the nephron was lost, and that this loss of nephron was also reducing the amount of podocin protein present. So that was using a patient 2 ml blood sample to make their own organoid. In a different approach, we've collaborated more recently with Amo Majumdar and Friedhelm Hildebrand to actually have a look at a novel gene that Friedhelm's group identified, NOS1AP, where they had a variety of different evidence that this was causative, and we engineered a knockout of this protein gene in a stem cell line and actually modelled the glomerulopathic disease. But today I'm going to talk to you about nephrotic syndrome and focus on MPHS2, the gene that encodes podocin. And this is work that's ongoing with ordoricin in my group. And what has been shown is that this protein that holds together the slit diaphragm complex at its base can be mutated in children presenting with nephrotic syndrome. A wide variety of single base changes can encode missense mutations right along the length of this protein. So ordoricin really wanted to have a look at what this meant. Corinne Antoniak had proposed by overexpressing mutations that these were mistrafficking, but what we did was go in and use CRISPR-Cas9 gene editing to actually edit the gene itself in stem cell lines, a control stem cell line. We created an allelic series where we made 5 distinct point mutations that encoded clinically relevant missense mutations in this gene, and then we made kidney organoids. These happen to be stripes, not dots, but within them you can see patterning and segmenting nephrons. So the presence of the mutation did not stop the formation, But what we found when we looked at the organoids is while in the control you can see nice polarized expression of podocin, in all these mutants the protein levels were substantially reduced, so the protein is being degraded. If you look at higher intensity fluorescence, you can see podocin protein. Here's our control glomerulus, but you can see here that these are diffuse and actually completely mistrafficked, and the specific mutation dictates where that trafficking is going to. So we have some stuck in the ER, some stuck in the Golgi, some in recycling endosomes, and it depends upon the mutation where the mistrafficking is occurring. One of these is quite interesting because we'd not seen a protein like this. Here's in the control at the basement membrane, but here you can see the protein stuck in the GM130 positive Golgi. And we actually were fortunate enough to have a patient, and we could show that in a patient line, which is a homozygote mutation, versus the parent, which is a heterozygote, we again saw this inappropriate localization in the Golgi. In collaboration with Jesper Olsen, we asked, what is the impact of this and why is it in the Golgi? And by looking at the control versus the het and the homozygote, we could show that the mutant peptide was only present in mum and the patient, and that the homozygote segregated quite separately with a very distinct proteomic profile. And when you look at hom versus het or hom versus wild type, we could see these distinct trafficking proteins that were affected in the patient only. What that showed us actually was that we're getting an impact on a lot of these molecules that carry cargo between the ER and the Golgi, but we're also seeing increased endosomal recycling of this protein. So the protein is probably being trafficked out to the membrane, failing to dock and being moved back. In the process, it's being degraded. So we're very excited now that we want to be able to take some of these mutations in podocin and take them further. But can we actually screen for compounds with such a complex tissue? This is a really complicated structure. It has many glomeruli. It's difficult to image. It's expensive to make. But what we have here is a nice readout. In the presence of a mutation such as this common R138Q mutation, you see a halving of podocin protein. And Ord has actually modified the protocol so that she can now actually not make whole nephrons, but make these beautiful little pumpkin-shaped glomeruli as a lawn across a 384-well tray that contain these gorgeous patterned podocytes in glomeruli, and we're actually starting to use these both for drug screening and for actually looking for AAVs that are specifically trophic for the podocyte. So what about the proximal tubule? This is a real target for nephrotoxicity and infection, but really it's a very complicated structure, and the mature proximal tubule has an S1, S2, and S3 segment, along which there are really distinct functions, and a mature proximal tubule is decorated with many, many solute transporters. What we have in an organoid is a model of a very early developing kidney. So Jess Van Slambrook in my group really wanted to ask, can we make this better? And you may have seen her poster during the week. What she said is, I would like to expand the nephron progenitors and pattern them more correctly. And she used a media that had been published from Shunsuke Tanagawa, who suggested that this media would promote the nephron progenitors, the stem cells of the nephrons. And what she found is by extending this protocol and adding this media, she could substantially increase 6-2 expression, which marked those progenitors, and give a more posterior Hox code, which suggests we're actually in the right part of the developing embryo. These organoids are stunning. They have this beautiful sundial array where now the nephrons are all lining up with the glomeruli in the circle and the proximal tubule extending away from the center. Using a HNF4 alpha reporter, which is a marker of the developing proximal tubule, we could substantially increase from about 4% to about 30% the number of proximal tubules present. And those proximal tubules, compared to a standard organoid on the left, are upregulating a lot of the solute markers, solute channels, and expressions patterns that we would expect to see in a more mature proximal tubule, including evidence of patterning to S1, S2, and S3 segments. In comparison to a standard organoid, these take up albumin very efficiently. You can see this beautiful take-up of the labeled albumin, and they also transport cations, and that cation transport can be blocked by cimetidine. Finally, they actually show much better expression than a standard organoid of many of the transporters that are involved in cisplatin influx and efflux, and in response to cisplatin, you can see this beautiful KIM1 upregulation along the proximal tubule. Because we were in lockdown, we actually asked, will they actually be infected by SARS? Because they actually show ACE2 expression and show upregulation of many of the other compounds that are important for entry of the virus into cells. We also found that the viral infection was much better in these organoids. ACE2 was expressed along the proximal tubules, and virus was actually entering those proximal tubules, but not the podocyte, which is what we should expect from pathology. If you knock out ACE2, you can make beautiful organoids, but in the absence of ACE2, you cannot get viral uptake. And we looked at both the endocytosis pathway for entry of SARS-CoV-2 into cells, as well as the cell membrane fusion pathway by inhibiting each of them with specific inhibitors, and found that, in fact, both inhibitors together are synergized, and so the virus is using both pathways. Importantly, TMPRSS2 is not expressed in the proximal tubule, and we believe that it's TMPRSS10 in the kidney that's working. So I hope that shows you what we've been able to do, some of the examples of what we've been able to do with organoids, but I want to answer the question of, can you really make a tissue? And that's because we're facing this kidney failure challenge, which is really affecting large numbers of patients, a growing number of the population, whereas we're still not increasing the availability of organs for transplant, which is the most optimal treatment. And really, treatment for end-stage renal disease has not substantially shifted in modality for a very long time. So let's ask some questions. I can say right now, we can't treat your patients with an organoid, but let's ask, what might we be able to do? So what does a transplantable auxiliary kidney need to deliver? Well, first, it needs to have complex microtissue containing nephrons, which I've shown you. We also need to make sufficient nephrons. I would remind you that a human kidney has on average 1 million nephrons. Even a mouse has 16,000, and the microtissue I showed you has 100. So we have a long way to go. And in the laboratory, we've been taking two approaches. This is an extrusion bioprinting approach, where we're actually printing the intermediate mesoderm onto a transwell filter. And now, instead of making a small structure, we can make large sheets with thousands of nephrons. And this has been shown to be able to be scaled up to tens of thousands of nephrons, too large for us to transplant into a small animal model. The other approach, which is somewhat more appealing from a manufacturing point of view, is to go down in scale and use bioreactors to make large amounts of material. And so Santosh Kumar, who's actually at this meeting, showed you could actually make microorganoids. Now we've got 5 to 10 nephrons, and these can be grown in suspension culture and so scaled up to a very large degree. These are fabulous for quality control. You can essentially very rapidly test their sphericity, their diameter, and you can image them all the way through so you can see whether the patterning is appropriate and the tubules are forming well. The next thing we would need is vascularization post-transplantation, and in 2018 we showed with Ton Rabeling using some of our fluorescent reporter iPS cell lines that when you place an organoid under the renal capsule, you get really rapid vascularization. So here's actually the human tissue in section stained with a red antibody to the nucleus of human cells, and you can see this MECA32 mouse endothelium has grown into the organoid and formed a beautiful vascular network in this glomerulus. Here you can see that glomerulus, which is blue because it's human and has a fluorescent reporter. And by putting a dye into the vasculature of the mouse, you can see that there's patent flow through this glomerulus and actually beautiful maturation of the glomerular basement membrane and the foot processes of the podocytes. We can also show that the vasculature that we put in there, which was human and is marked in red because of SOX17, is anastomosing, and we're forming a patent vasculature that includes human endothelium. And in a really beautiful study, Katalin Vandenberg actually put in both large and small molecular weight dextrans colored with different fluorophores and showed that the large molecular weight dextran in green stayed in the vasculature and the small molecular weight dextran crossed the basement membrane. In fact, they calculated that the size selective sieving was appropriate compared to human tissue. The final thing we need is a patent connection between the transplanted nephrons and the host renal tubules. And I'd really acknowledge the work of a number of groups, particularly these two, and this is Min Chi and Carl McCracken, who's in the audience, and this is a work in Ryuichi Nishi-Nakamura's lab, where they've been focused on making the collecting duct network itself. We're taking a different approach. We're actually transplanting the tissue itself as a body of nephrons. And we actually have shown, as I've illustrated previously, that the vasculature grows in very rapidly. Within 7 days, you get vascularization of the glomeruli. What we really want to do is see if we can induce this connection so that the nephrons we transplant are essentially hitchhiking on the collecting duct system of the patient. And Kynan Lawler in my group is driving this team. It's a very laborious process because we're doing this very quantitatively. Every tissue is processed into large, thick sections. We have a variety of different staining panels looking at the number of glomeruli, whether they're vascularized, the distal tubular content, and we essentially confocal stack all of these sections so that we can have a look at this. Here's a transplant marked up for the distal tubule and the glomeruli of the graft itself. And here's another graph that I'll show you in a little bit more detail. What we're really looking for is evidence of connection between the host and the graft and a robust connectivity. And I would say we're not there yet, but we're looking very hard. So I hope in that short period of time that I've been able to run through a decade of stem cell-derived kidney tissue that I've shown you the variety of approaches that have been used. We started with this micro tissue that has many of these patterned and segmented nephrons. We're now specifying specifically to the glomerula so that we can do drug screening. We've actually shown that you can scale up either by making large numbers of microorganoids or by making sheets using bioprinting. We've also shown that you can improve the patterning of the proximal tubule and that Jess is now working on getting these proximal tubules to align. And we've also, and I haven't had time to talk about this, shown that we can make the collecting duct and get it to fuse to the nephrons. And in work that's unpublished, we're now stringing these together to make kidney structures. So I'll stop there and thank my fantastic team and the many people that we've collaborated with over the years, some of whom are in the audience. And I particularly thank all my staff, Jess and Ord, who are here, Kynan, who's working on the transplant work, Sean, who does all the transcriptional analysis, Sarah Houghton, who does all of our gene editing, many funders that have supported this work over the years, the many students, the many colleagues and collaborators in this organoid field, and I thank you all for your attention.
Thank you very much, Dr. Little, for your outstanding state-of-the-art presentation. This plenary session has concluded. Please visit the last day of exhibits and posters. Thank you so much.